Thank you, Chairman. Hello, everyone. Mucho gusto, encantado. Me llamo Takummi Hosono. Thank you. I'd like to talk about my research on isolation and purification of decorin from bovine skeletal muscle and its structure changes under high pressure. First of all, I'd like to talk about outline of my presentation. First, background. Next, isolation and purification of decorin from bovine skeletal muscle, and changes in structure of purified decorin and high pressure. Finally, summary. Background. What is the definition of high pressure? 0.1 megapascal. This is atmospheric pressure. Normally, we live under atmospheric pressure. Deep sea organisms live under pressure greater than 0.1 megapascal. As pressure increases, these phenomena occur. I focus on these phenomena at 300 to 400 megapascal. Protein denaturation occurred, followed by protein precipitation. I apply these phenomena to meat protein structure. Next, I'd like to talk about effect of high pressure on meat protein structure. About actin-myosin toughness, Suzuki et al. Have reported that modification of actin-myosin interaction, fragmentation of myofibrils, and conversion of alpha-connectin to beta-connectin. In addition to these reports, there are various reports about effect of high pressure on actin-myosin toughness. About background toughness, Icomst et al. Have reported that collagen molecules do not degrade, but collagen fibrils dissociate by high pressure. However, about background toughness, this report is only published once since then. That is, detailed mechanisms about effect of high pressure on background toughness have not been clarified. So I think that it is necessary to clarify these mechanisms. About major component of connective tissue. As you know, meat is composed of various proteins. Collagen fiber is main component of connective tissue. Collagen fiber is composed of collagen fibrils, and collagen fibrils is formed by the association of collagen molecules. Decorin, which I have studied, is this. Decorin is small proteoglycan with arch-shaped structure and is composed of a core protein and one glycosaminoglycan chain. One of the key roles of decorin is to bind and interact with collagen. At previous slide, I explained that collagen molecules do not degrade, but collagen fibrils dissociate by high pressure treatment. Based on this report, I assume that decorin conformation is affected by high pressure. That is, decorin conformation changed by high pressure leads to destabilization of collagen fibril structure, and it results in weakening of connective tissue, eventually leading to meat tenderization. So, in the present study, decorin was isolated and purified from bovine skeletal muscle, and then structural changes of purified decorin were analyzed by fluorescence spectra under high pressure. Isolation and purification of decorin from bovine skeletal muscle. First, the process of isolation and purification of decorin was shown on this screen. First, decorin was extracted with formula guanidine hydrochloride and cesium chloride density gradient ultracentrifugation from bovine skeletal muscle. However, it contained unnecessary ingredients such as hyaluronic acid and so on. So we applied DEAE cellulose ion exchange chromatography and cephalo-CL6B gel filtration chromatography to remove the unnecessary ingredient. Recovered samples were analyzed by SDS page to characterize as decorin with Kumashi brilliant blue, Alcian blue, and silver staining. Decorin through these steps was used for measurement of fluorescence spectra. The results of ion exchange chromatography. Proteoglycans were eluted with linear gradient of 0 to 0.5 molar NaCl, shown by green line. And blue line indicate protein content. And red line indicate proteoglycan content. The fraction 27 to 32 contain decorin with molecular mass of 100 kilodalton by SDS page and were combined. And the gels were stained with Alcian blue. These fractions were concentrated to about 10 milliliters by ultrafiltration and were subjected to gel filtration chromatography for further purification. The results of gel filtration chromatography. The fraction 18 to 26 contain decorin with molecular mass of 100 kilodalton by SDS page and were recovered. And the gel was stained with silver staining. Recovered samples were combined and freeze-dried. And freeze-dried decorin was used to next experiment. Next, SDS page of purchased decorin from bovine cartilage, and purified decorin from bovine skeletal muscle, and purified decorin from bovine skeletal muscle at 400 megapascal pressure was shown on this figure. And the gel was stained with Alcian blue. SDS page revealed that decorin from bovine skeletal muscle was not degraded by high pressure, up to 400 megapascal. Next, changes in structure of purified decorin under high pressure. The process of measurement of fluorescence spectra for purified decorin under high pressure was shown on this screen. Fluorescence spectra indicate changes in tertiary structure of protein. First, 0.2 milligram per minute of decorin solution was prepared with 15 millimolar toric acetate buffer. And decorin solution was injected into the high-pressure cuvette. The high-pressure cuvette was set in pressure vessel. With a hand pump, the vessel was applied to pressure up to 400 megapascal in stages. After maintaining each levels of pressure for 10 minutes, fluorescence spectra were measured under high pressure. And fluorescence spectra of purified decorin were measured with excitation at 280 nanometer. Next, I will explain that changes in fluorescence spectra of purified decorin under high pressure. Fluorescence spectroscopy was used to analyze the environment of peripheral tyrosine and tryptophan residues in native and denatured decorin. About compression, this value is fluorescence intensity at 0.1 megapascal. As pressure increases, a reduction of intensity and shifting peak emission wavelengths, red shift, were observed. These results indicate that the tyrosine and tryptophan residues of purified decorin were exposed from interior to polar environment that is outside by high pressure, and decline was denatured. About decompression, this value is fluorescence intensity at 400 megapascal. With pressure decrease, fluorescence intensity and peak emission wavelengths return to native decline value, blue shift. These results indicate that high pressure induced changes in tertiary structure of purified decline reversibly. Next, center of spectral mass was calculated. This fluorescence spectral value. Next, changes in center of spectral mass of purified decline under high pressure. Center of spectral mass was used to indicate changes in the approximate shape of protein, and changes in the environment of peripheral tyrosine and tryptophan residues of purified decline leads to changes in center of spectral mass, new value. New value was calculated by this formula. About results, the new value decreased gradually with increasing pressure level. Furthermore, with pressure decreased, the new value returned to initial value. These results also indicate that high pressure induced tertiary structure of purified decline reversibly. Therefore, the present results indicate that decline from bovine skeletal muscle is not degraded. However, the tertiary structure of decline is changed reversibly under high pressure up to 400 megapascal. Finally, I'd like to talk about summary on my research. Why meat tenderization is occurred by high pressure treatment? Weakening of connective tissue is considered as one of the reasons for this question. First, high pressure affects decline structure, causing destabilization of collagen fibrils, and it results in weakening of connective tissue, leading to meat tenderization, I think. Extension of these mechanisms would be beneficial to the development of new food processing technology in meat product. If you have some question, please more slowly in English, because I'm a student, so my listening ability is very poor. Thank you for your kind attention. Gracias.